Okay. All right. Hello, everyone. We are thrilled to have you here this evening for the second session of the Cedar River Salmon Journey webinar series. My name is Charlotte Spang, and I'm here with Danny Kendall. And together, Danny and I work on the Seattle Aquarium's field programs, which include the Cedar River Salmon Journey and the Beach Naturalist programs. We're very happy to have you here this evening, so thank you for joining us. Before we get started, I'd like to acknowledge that we are on the traditional lands of the Coast Salish people who've cared for these lands and salmon since time immemorial and continue to do so today. Washington State tribes are co-managers of our state salmon with the Washington Department of Fish and Wildlife. We hope you enjoyed last week's introduction to the Cedar River watershed with Katie Klon, and that you'll join us for our final session next Tuesday when Jason Mulvihill-Kuntz, who is the salmon recovery manager for the Lake Washington Cedar Sammamish watershed, also known as RIA 8, will speak about efforts to restore salmon in the watershed and what we can all do to help. For those of you who were not able to attend last week, we will be posting the sessions to the Aquarium's YouTube channel, so let us know if you'd like to know how to access those when we get them posted. We also encourage you to get outside and see salmon. They are spawning in the Cedar River right now, and we are spotting salmon, both sockeye, which we've seen at every single one of our 6 sites along the Cedar River, and we've seen Chinook at 2 of our sites. So I think at your earliest opportunity, when the weather clears, you might want to give the river a couple days to clear. It's probably pretty turbid right now after all this rain. And if you don't live near the Cedar River, perhaps check out a local stream or river near you. It's really amazing to be outside and be able to see these phenomenal fish that we get to share our watershed with. We are grateful for the amazing support from volunteers, partners, and funders that has allowed the Cedar River Salmon Journey program to reach thousands of people every year since 1998. This year's program is funded by a cooperative watershed management grant through the King County Flood Control District in partnership with the Lake Washington Cedar Sammamish Watershed Salmon Recovery Council. We also receive funding from the MMS Foundation, and we're grateful for the support. So many thanks to all of you. For tonight's presentation, we will be hearing from Aaron Bosworth, who is a district fish biologist with the Washington Department of Fish and Wildlife. He monitors salmon and steelhead spawning escapement in the Green River and Lake Washington watersheds. Aaron's work group also helps monitor hatchery programs and set fishing seasons in these watersheds. He's been working in this position since 2009. Aaron is going to be speaking about salmon of the Cedar River, how they're doing, and population trends. We will be taking questions at the end of his presentation, but please use the Q&A function at the bottom of your Zoom screen to ask questions anytime. I'll read them to Aaron at the end of his talk. Please reach out to us via the Q&A if you have any trouble, or email salmonjourney@seattleaquarium.org if you are having any trouble or can't communicate via the Q&A. Aaron, thank you so much for joining us this evening. We're really thrilled to have you back speaking with us about these fish that we love so much, and we are really looking forward to learning more about them from you this evening. So thank you, Aaron, and welcome. I'm going to stop sharing my screen so that you can start. But in the meantime, while you're getting ready to do that, again, I just want to encourage folks to get out to the Cedar River. It's such a beautiful place to watch and observe salmon, and our sites are at 6 different locations along the river, and it's a fun place to explore and to see salmon in a natural habitat, and also to see how salmon can do so well in an urbanized area as well. So Aaron, thank you, and take it away.
Okay, thanks again, Charlotte. Can you hear me okay?
Yes, I can hear you wonderfully. I'm going to go ahead and mute myself now.
Okay, great. And just to echo what Charlotte said earlier, yeah, if you want to see salmon, go out to the rivers soon. I think the peak of our spawning season probably just passed us by maybe last week, and I think that the numbers of fish spawning in the rivers are probably going to decline from this point till the end of October and into early November. So go out there soon if you haven't yet, see the salmon. So again, yeah, I'm going to talk about salmon in the Cedar River. Here's a short outline of some of the things I want to talk about more specifically. I like to start by talking about the history of the Cedar River. Then I'm going to talk about the salmon in the Cedar and some of the other fish that live there. And then people always wonder, how do you know about the salmon? So I'm going to talk about some of the monitoring programs that we conduct in the Cedar, and then finally some of the monitoring programs that we conduct or help with rather down at the Ballard Locks. So that's kind of a short outline of the things I want to talk about. Let me just go right into the history of it. This is going way back around 17,000 years ago, when the entire area was covered with ice, and of course there was no Cedar River back then. This is the Puget Lobe of the Cordilleran ice sheet. And then as that receded, well, before it receded, it's important to note there was a lot of ice over this area, and it had a pretty profound effect on the landforms underneath it. And as it receded, well, this is the area that it covered here, and after it receded, it had left all these kind of north-south oriented landforms, and I just love to think about how these waterways were formed by the glacial ice sheets. And so this one in particular, I hope you can see my mouse as I move it around here, but this is where Lake Washington is, and here's Lake Sammamish, and then up here is the headwaters for the Issaquah drainage. So that Issaquah Creek comes down into Lake Sammamish, and then it gets into the Sammamish River and follows this northerly route up and curls around until it comes into Lake Washington. And then historically it would flow this way out what used to be called the Black River, which was down here, and then it hooked around and joined up with the Green Duwamish River, and then it flowed back north and up into Elliott Bay. And that's how the watershed worked. And here was the Cedar River coming down out of the hills, and it joined not into Lake Washington, but into the historic Black River. And it also went out into the Duwamish waterway. So a lot of people have heard that story, but I just kind of like to review that fact that the Cedar historically didn't flow into Lake Washington at all. And so here's a cool picture of what Lake Washington probably looked like prior to the time when the Cedar was rerouted. So up here, this is the Sammamish River coming into the north end of the lake. And historically, there wasn't an outlet at the Lake Washington Ship Canal. And all the water flowed south to the Renton and entered the Black River here. And this is the historic Black River, which flowed, it wasn't a very long river, but it flowed down and into the Green Duwamish and then out into Elliott Bay here. And then this is the historic Cedar River coming into the Black River. And here's a close-up of that area, that Renton area. Again, here's the Black River coming out of Lake Washington, the Cedar River flowing into the Black, and then the Black goes into the Duwamish waterway. So that's the way things used to be. And I like these historic photos, so I just thought I'd put a couple of these in here. This is the historic Black River, what it looked like. So it was kind of a wide, slow moving, kind of a low gradient affair. If you could see here, there's rowboats and canoes were able to go up and down the river. So it wasn't a raging river by any means, but it was a pretty significant river back in the day. And here's another one, all these pictures down around the Renton and Tukwila area. But then around 1915, 1916 is when the Cedar was rerouted. So again, on the left-hand side of the screen is the historic configuration with the Cedar coming into the Black River and out to Elliott Bay. And then it was rerouted 1915, 1916, and the Cedar went right into Lake Washington, and the Black River at that time had ceased to exist. And of course, not shown here is the Lake Washington Ship Canal, and that became the new outlet for the Lake Washington Basin. And so more historic photography here. Here's the crew of people that was rerouting the mouth of the Cedar down in the Renton area, rerouting it into Lake Washington and out away from the Black River. And then here's what had to happen at the Lake Washington Ship Canal to allow all that to happen. Of course, the Ballard locks were constructed, and then the Fremont Cut and the Montlake Cut had to be constructed for this area to be the new outlet for Lake Washington Basin and the Cedar River. And here's some more photos I like. I've shown these at other talks, but I'd show them every time. I like them. This, I think this is the Montlake Cut when it was finally being opened up. And here in the background is Lake Washington. And Lake Washington was lowered 9 feet when that cut was let go. So here's all the water kind of rushing through this what used to be the dam. And here's the next photo. And it must have been quite an exciting event to be at. And looks a little dangerous to me, actually. And then this final shot is the Black River after that happened, and it dried up, and there's historical stories of fish being trapped in those pools of water and people going and netting them out, and then kind of ceased to exist after that. So that was the end of the Black River. Well, okay, let me move into the fish portion of the presentation. So the Cedar River, historically, when it went into the Green River, it certainly had Chinook in it and coho, and it probably also had chum salmon, pink salmon, and steelhead. It probably had all of those species in it. But when it was rerouted into the Lake Washington Basin, coho, or not coho, but chum salmon and pink salmon couldn't survive in that river anymore. They don't like watersheds where there's a big lake between the spawning areas and where they get out to marine waters. But Chinook still persisted in the Cedar River, and coho did too, and it created a new opportunity for a new species, sockeye, which were introduced from Baker Lake into the Cedar, and now they're the most abundant salmon species in the Cedar. So these are kind of the three primary salmon species inhabiting the Cedar River today, and these are the ones that I'm going to talk about in the talk. So again, there's Chinook in the Cedar, there's sockeye, and there's coho, and those are the big three salmon species that inhabit the Cedar. But by no means are those the only species. There's a lot of species of fish in the Cedar River, so I wanted to mention a few other ones. Many people don't realize that there's a bull trout population that inhabits the Cedar River. It's kind of a landlocked bull trout population, and it lives exclusively in Chester Morse Lake up in the municipal watershed. It's not a big population, and you probably won't see a bull trout in the lower river. We never see them, but there's a small stable population up there. And down here in the lower right, we have steelhead. Steelhead were once abundant in the Cedar, even after it was diverted. But the Department of Fish and Wildlife used to have large hatchery releases there also. And then over the years, the hatchery practices were discontinued for steelhead there. And the numbers of anadromous steelhead coming back to the Cedar declined, and there basically is no more steelhead using the Cedar, maybe between 2 and 5 fish. And in some years we don't detect any fish. And we look for steelhead, we conduct surveys to see if they're spawning there, and we don't find them every spring. So steelhead are not using the Cedar River right now. However, rainbow trout, which is kind of a resident form of the steelhead, are relatively abundant in the Cedar River, and so are cutthroat trout. And so these fish species inhabit the Cedar, and in fact, there's a short fishing season in the summer, and people like to go up, and it's a catch and release fishery, and they fish for those species. And so not to leave out some of the other species that live there, sculpin are quite abundant in the Cedar River, and they occur throughout the river. And then there's a really healthy population of longfin smelt. It's kind of a special little population of fish that actually they live most of their life in Lake Washington, but they migrate up into the lower Cedar to spawn. And then here's a brook lamprey. These are a cool little species. There's lots of those in the Cedar. And there's a bunch of other fish species that I'm not going to show here. There's whitefish and large-scale suckers, other fish species in the Cedar beyond the salmon that we all like to talk about the most. Oh, and here's one last fish species that sometimes occurs in the scene in the Cedar River. This is a sturgeon, and a little known fact, sturgeons sometimes come into Lake Washington, and I think they hang out there for long periods of time, and they've been seen in the Cedar on certain occasions. And this is a historic photo of a really big sturgeon that was caught in Lake Washington, I think down near the Renton area. This other picture was one somebody sent me. This is actually taken up near Kenmore, but these sturgeon are in Lake Washington. They sometimes go up into the Cedar too. So kind of exciting to think about these big fish around the Cedar River. So here's a few photos of the Cedar River. This is kind of the upper portion of the river where I spend some time doing fish surveys. Really pretty up there if you haven't been up. And there's these kind of big walls with gravel and cobble sticking out, and these are constantly bleeding down into the river. That's kind of a good thing. It recruits nice spawning gravels for the salmon off these walls. But my one word of caution is if you're a tuber, and I know there's lots of people that like to inner tube on the Cedar, kind of stay away from these tall walls, because the cobble and the gravel can come out of those at any time and come down. It would be a bad thing to get hit on the head by one of those things. So just some pictures I took from earlier this fall when I was on surveys out there. So this is a kind of typical habitat in the upper parts of the Cedar where we do our stream surveys. So let me talk about just Chinook here and some of our Chinook monitoring programs in the Cedar, and some of the abundances that we've been finding. So in 2019, we're nearing the end of the spawning season for 2020, so I won't share any numbers from this year, but this is what we found in there last year. So it was about 855 Chinook that spawned in the Cedar River last year. That's what we estimated. And here's the trend of that over time. If you can see, it kind of varies in time. At its peak, it gets up over 2,000 fish for Chinook in the Cedar, and it goes down to about 500. And so I guess what you can take home from this is that it's not a large population of Chinook that lives in the Cedar, but it's relatively stable. It has its small fluctuations, but never gets super low. And last year's spawning escapement was maybe on the low end, but still around average, I would say. So not a bad population of Chinook. And the way we determine how many Chinook spawn in the Cedars, we float the entire river. When I say we, it's kind of a group project. A number of different agencies participate in these surveys. It takes a lot of people, but we do what's called a complete red census, and we look at Chinook reds, and we think we count every single one of them that's constructed throughout the spawning season, and that's how we determine the Chinook abundance every year, and spawning distribution within the river. So this is one of the rafts that we use and float down the river. And many of you have probably seen Chinook reds before, but I like to put a few pictures of actual Chinook reds in here. And this actually was taken in the Green River from a helicopter. And these are all Chinook reds, these light covered areas. Now the Green River is a little different than Cedar. The algae covers most of the rocks in the Green River. So it has this green appearance. And when a Chinook creates a red, all of these are multiple reds, it cleans that algae off the rocks, and the red is really easy to see compared to the gravel that has the algae on it. So the reds really show up well, and you can actually see these are actual fish on the reds. And I don't think all of them are Chinook. There may be some pink salmon mixed in there in this particular area. But these are what Chinook reds look like, and they're quite large too. And here's another closer photo of a red in the Green River. Also, you can see it has this greenish tinge to it, and there's a little bit of algae on the rocks. And then this is just a bright spot, and you can see a Chinook has dug a pit here, and then the tail out is here. There's kind of a mound of gravel, and there's pockets of eggs covered by this mound of gravel. And there's a little piece of green flagging here in the Green River. Sometimes we throw flagging in on the red so that we can tell from week to week that we've already counted that red, and we don't double count it. So it's a way of marking that red. Now here's a photo of Chinook red from the Cedar River. The Cedar River is a little more challenging to identify reds. If you've seen them there, the substrate in the Cedar River is a little more clean than in the Green River. There isn't all that algae on the rocks. And so obviously these are sockeye in this picture in the foreground, and they're constructing reds here too. So this gravel's been cleaned off. But if you see this larger fish, this olive colored fish with a dark and and a little bit of white on its tail. That's a big female Chinook, and she's constructing a red right there. And so the difference here is that that red, the substrate beneath it really isn't that different in color from the surrounding substrate. So it can be a little tricky to spot a Chinook red in the Cedar River. And so I like to show that picture to show that these Chinook kind of really blend in. Oh, and here's another Chinook red. I took that this year. This is kind of in a side channel in the upper portion of the Cedar River. So this is a Chinook red here. That's the fish is actively digging, and here's the tail out here. And I don't know if you can see or not, but there's a big female Chinook on this red in the picture. And it's, what they often do is there's no cover, and so they'll just hold real still right on the bottom and not even move at all. And it's really difficult to see them. But here where I'm circling the fish with my cursor right here. So that's that big female Chinook on that red. Here's this next picture shows a closer look at her. So that's the same red, and here's the female Chinook. And if you look at her tail, you can see this white starting to form, and we call that a flagtail Chinook. And what happens as she digs the red, there's an abrasion on her tail from it banging against this substrate down here, and gradually the female Chinook's tail will turn all the way white and almost and erode down to this little nub. And so that's one way we can tell if a female Chinook successfully dug a red, is if she has that flagtail. So this is a fresh Chinook, and her tail is just starting to turn into that flag color. So anyway, that's kind of interesting. So now I want to talk about another monitoring program that we do in conjunction with the red surveys. We also do carcass surveys for Chinook, and we get a lot of information from these carcasses. As everyone knows, the Chinook all spawn and then they die, and then their bodies just kind of lay around and float to the edge of the river. And as we float down, we look for every single one that we can find. They disappear kind of quickly, and we sample these carcasses and we get a ton of different information out of the carcasses. So we do carcass surveys in there also. And I just love these carcass pictures. I just had to share a few with you. And it's a part of fall for me. I like the leaves changing colors and the days getting a little cooler, and the Chinook kind of spawn and then they die, and then their carcasses start to rot, and it's all part of the same thing. They kind of get this ghoulish appearance. It puts me in the mood for Halloween always. So this time of year is when most of the Chinook are dying off. So it's the peak of our carcass survey. So we're out there getting these carcasses like crazy and sampling them. And these are relatively fresh carcasses. It's nice when you can find a Chinook carcass pretty soon after it dies, because it's pretty good to handle. But if you look at some of these in the lower right or upper right, these Chinook have been around for a few days, and they start to fungus up pretty quickly. And then here's some other carcasses, and we also have to sample these. If you don't get them right away, if you let them sit around for a week, they start to look like this. And I just love to take pictures of these old rotting carcasses. This one's just one of my favorites here. Actually, I think this is a sockeye carcass, but you can see all the maggots coming out of this fish. Really kind of a good Halloween scene. But we sample all these carcasses, and this is how it goes. We'll find a carcass in the stream. This is one of the members of our stream survey crew. We usually get them on a hook with a long pole, and we'll take them and we'll fix them to the boat that we're floating down the river in, and we'll put, well, as many on there as we can fit, usually 5 or 6 at a time, and take them down just for efficiency's sake. And then when we get a good batch of them, we line them up on the bank, and then we start to collect the information from them all at once. So it just makes things go quicker. So here's some big collections of carcasses that we've had in different years, and we'll line them up like this, and then get a couple of us and sample them after that. Here's one of my technicians sampling this Chinook. Some of the information that we get, we do get a length from them, and we get the gender, whether it's male or female. We look at the adipose fin to see if it's a hatchery origin fish or a natural origin fish. And then this person has taken scale samples right here. We always take scale samples from the flank of the fish, and we put them on this adhesive scale card in this book. And we typically use the scale samples to determine how old that fish is. So we'll take usually 6 scales from every fish, and then fill these cards up and send them to our lab, and they'll tell us the age of those fish. If you look up in the upper right here, sometimes for some fish, we'll extract what's called the otoliths out of the brain cavity. And so that's what this person is doing here with a little hacksaw. And the otoliths are two calcified structures that kind of float in the brain cavity on either side of the brain. And so we'll cut in there, and then we pull the otoliths out. And those, we can get a number of pieces of information from the otoliths. One thing is the fish's age. And then another thing is whether it's a hatchery origin or a natural origin fish. And there's some other pieces of information that people like to get to. Let me move to this lower right picture. This is a female Chinook. And one thing we look for in female Chinook is whether they've spawned successfully or not. So we open up the body cavity to see how many eggs are left in there. And this one, you can see there's a handful of eggs, but really not that many. And so we think that this Chinook probably spawned successfully, but there's some Chinook and they have all of their eggs left in them. And what's happened, that Chinook has died before it had a chance to spawn, or it's experienced what we call pre-spawn mortality. And that's kind of a bad problem for an adult Chinook, to make it all the way back to the river, but then not have the energy to spawn before it dies. And so we like to keep track of that. And luckily in the Cedar River, we have really low rates of pre-spawn mortality, meaning that most of the Chinook we find are like this, or even more of the eggs are gone than this, I would say, in most Chinook we find on the Cedar. They for the most part, spawn successfully. And so here's a summary of some of the information that we get from those carcass surveys and what we found for the Cedar River. So in 2019 in Chinook on the Cedar River, what we found was that about 70% of the fish that spawned there were natural origin spawners, and about 30% were hatchery origin spawners. Now, hatchery origin spawners, there is no Chinook hatchery on the Cedar River. The only Chinook hatchery in the Lake Washington Basin is at Issaquah. So these hatchery origin spawners are fish that were produced at the Issaquah hatchery, and then as adults, they've strayed to the Cedar River and spawned there. And so we like to track that percentage on an annual basis. And here's that percentage going back into time. For quite a few years. We always keep track of this. What you can see from this is the orange colored bars on the bottom are the natural origin spawners in the Cedar, and the lighter colored bars at the top are the hatchery origin spawners. So what you should see from this is that most fish in all years in the Cedar River are natural origin spawners. 2014 is kind of an outlier when close to half the fish were hatchery origin spawners. And in recent years, the number of hatchery origin spawners has gotten a little greater than it used to be back in these years. But overall, most of these Chinook are natural origin fish that are spawning in the Cedar River. And so here's some information from those scale samples. It tells us that about 70% of the fish we saw in 2019 were 4-year-old fish. And then about 27 were 3-year-old fish, 2% were age 5 fish, and about 1% were age 2 fish. So that was the age composition of the fish that returned in 2019. And this graph tells you that that's kind of the norm. What we normally see is that Chinook that spawn in the Cedar River are age 4 fish, or in some years age 3 fish predominate. So these darker bars represent the age 4 fish, and you can see that in many years there's a lot of age 4 fish, and when there isn't, usually age 3 fish are the dominant age class that we see. And then down here in the red ones are age 2 fish. You can see not a lot of age 2 fish, and some people refer to those as jacks. They're much smaller fish come back. There's always some, but it's a very low percentage. And then up here, these tan color bars are the age 5 fishes, and there's also not very many of those either. Okay, I've been talking about the Cedar River. I just also wanted to mention the Sammamish population of Chinook. So when Chinook come into the Lake Washington Basin, they come in through the Lake Washington Ship Canal, and they can either go to the Cedar or they come up here into the Sammamish River, where a lot of them spawn in Big Bear Creek or Cottage Lake Creek, or they swim through Lake Sammamish and into the Issaquah River or return to the Issaquah Hatchery. And so that's characterized as the Sammamish population of Chinook. So those are the remainder of the Chinook that come into the Lake Washington Basin. And so here's a graph of the abundances of Sammamish Chinook over time. And over the last 20 years or so, this, really this population isn't too much different than the cedar. It reaches highs of around 2,000 fish and lows of around 500. So last year we kind of had a little bit of a lower return, if that's okay. So this population is kind of on par with the cedar in terms of its abundance. So these rivers up in the Sammamish, or creeks rather, are much smaller than the cedar. We can't float down them, and we walk all of these, do foot surveys, and that's how we figure out how many Chinook spawn in these different areas. And this is an example of one of the streams. This actually is Kelsey Creek in the golf course, but most of the streams in the Sammamish where the Chinook spawn are similar in size to this one, or even smaller sometimes. This is 2 pictures of Coal Creek that were taken this year. And this is kind of before water starts to rise in October, before we get the first rains. And you can see a Chinook would be challenged to even make it up through these little riffles. But as these streams rise a little bit, a small number of Chinook do come up, nose up into these small streams, and they'll spawn in some of these areas. And so for Sammamish Chinook, here's some of those same statistics I showed you before. Now Sammamish is the exact opposite of the cedar, actually. So about 70% of the naturally spawning fish are hatchery origin spawners. So that's opposite what we see in the cedar. And there's only about 30% of the spawners are natural origin fish. And if you look at that trend over time, you can see that's relatively consistent. Natural origin spawners are these smaller bars on the bottom, and then the lighter colored bars on the top are the hatchery origin spawners in the Sammamish population. So what that tells you is that most of those fish spawning in those streams are hatchery origin fish. So again, those are all hatchery fish that were produced at Issaquah Hatchery. They've come back as adults, but they haven't returned to the hatchery. They've strayed into Bear Creek or Cottage Lake Creek, or the lower parts of Issaquah Creek or other streams, and spawned there. And so those are naturally spawning hatchery fish. And here, I think this is a kind of a final graphic for Chinook in Lake Washington. These lower lines, so this dark red one is the Sammamish population. These are the abundances over time. And then this yellowish colored one is the cedar Chinook. And you can see these are kind of similar, cedar and Sammamish. And then this kind of orangish line that's above them is the number of fish that return to the Issaquah Hatchery. So you can see, if you look at all the Chinook that come back to the Lake Washington Basin, by far the highest abundance are the hatchery fish that are coming back to the Issaquah Hatchery. So it's good. That kind of puts things in a little bit of perspective. Okay, so that's all I'll say about Chinook. I'm just going to move on to coho, and I'm not going to talk about coho that much. They do spawn in the Cedar River, but they're difficult for us to monitor there. They don't spawn in the main stem areas, and they don't show up until later in October and November when stream flows are really high. And what the adults do is that, yeah, again, they don't like to spawn in the heavy water in the main stem. They nose up into smaller parts of tributary streams or way high in the watershed. It's really difficult for us to find their redds or find where they're spawning. So we do monitor a few indexes, and we see a few of the coho spawners there, but not a lot. So we don't have a good handle on the population that's actually spawning in the cedar. One of the best ways we track coho in the Lake Washington Basin is down at the Ballard Locks. So these are the numbers of coho that we've counted over the years. You can see 2019 is kind of a little bit of a low year, but not too far off of average. And again, here at the locks, these are fish. We count them as they go past the fish window, and we estimate how many are hatchery origin and natural origin. You can see the red bars are the hatchery origin coho, and the yellow bars are the natural origin fish. And so what you see there is that most of the fish coming back to the Lake Washington Basin, again, are hatchery origin fish. And that's because there's a large hatchery program at Issaquah. And so those fish are coming through Ballard Locks and probably not going to the Cedar. They're going to go back, most of them, and return to the Issaquah hatchery. Okay, and so let me move to sockeye. Everybody loves to talk about sockeye, and so do I. So cedar sockeye is probably the most abundant fish species in the Cedar. So in 2019, we estimated that about 17,000 fish sockeye came back through the Ballard Locks. Again, we count them there at the Ballard Locks, and then we count the number. We estimate the number of fish that spawn in the Cedar River. And in 2019, we estimated that about 3,500 sockeye spawned in the Cedar River. So those are our abundance estimates. And again, the way we get that, similar to the Chinook, is we float the whole river. With sockeye, we don't look for reds like we do with Chinook. We do what's called a live fish count, where we count the actual fish, and then use a calculation to estimate the total number of fish that had returned for the whole year. And so this is this is our setup, and we float the whole river. It's only once a week that we do that, on Tuesdays usually. So here's the number of naturally spawning cedar sockeye that we've seen. What you see is a long-term declining trend, and the sockeye population in the Cedar is kind of in a bad spot right now. In 2019 was the lowest spawning escapement we'd ever seen on record. I don't know if you remember, Charlotte mentioned that I have been working in this job since about 2009, and so at that time there was quite a steep decline, as you could see. And then after I started working, I really thought I was a successful fish manager, because the population just started to skyrocket. But then unfortunately it was not to be, because it crashed again in 2014. And despite my best efforts, it's been at a rock bottom state since then. So dashed all my hopes as a fish manager. But that aside, yeah, this population is not doing well right now in the Cedar. And so just a little bit about the monitoring program. So that I mentioned that we count the number of sockeye and Chinook and coho that come through the Ballard Locks. And again, they move up from marine waters. And many of you have been down here, I'm sure, and the fish viewing area is here in this corner, and the fish ladder. They have to enter the fish ladder. That's one way that they can swim up into the Ship Canal and get into Lake Washington. Or they come through the small locks this way, or they go through the large locks this way. And we think we do a pretty good job of estimating their numbers by counting them here and as they come through the large locks. And again, here's this is the fish viewing window, which many of you have seen, and some of the spillways. And this is a shot of the entrance to the fish ladder down there at the Ballard Locks, where we do some of our monitoring for abundance for sockeye. And so then we here's a graph. This shows the yellow outline shows the number of sockeye that we count at the Ballard Locks, and then the red one shows the number of fish that we estimate spawn. As you can see, there's more fish coming through the Ballard Locks than are actually spawning. The reason for that, some of these fish that come through the Ballard Locks are going out to spawn in the Sammamish River tributaries at North Creek, Little Bear Creek, and Big Bear Creek, but not that many. A lot of these fish go missing. Some of them get miscounted or overcounted at the locks, but we also think that some experience pre-spawn mortality in Lake Washington itself or in the Cedar River, and they die before they get a chance to spawn, and so they don't show up as natural spawners in the Cedar River. So you can see that there's quite a big difference in some years between what we see at the locks and what we count in the river. And this big red line is how many fish we have to see at the locks before we have a open fishing season for these sockeye. You can see the last time we did that was in 2006. And it's been some time since we've been able to fish for sockeye, since they've been abundant enough. Now let me talk about one more monitoring activity we do down at the Ballard Locks for sockeye. Similar to the carcass surveys, although in this one we actually capture some of the fish as they come through. And these are some of the tribal technicians. They're actually standing down in the fish ladder themselves. You can see they have their net right here. Well, what they do is they wait till some sockeye come into this chamber of the fish ladder. Actually, when they stick their net down in here, the fish viewing window is right there. So you can, if someone's watching the fish, they can see these fish getting netted out of there. But these technicians will net the sockeye out and pass them up to this guy that's sitting on the wall, and he'll then put them over into a tote in the boat on ice. And these are a bunch of the fish that we caught this year, actually, down at the ladder. And then the boat takes them up just a short distance to a dock where we process these samples. And we do all the same things that I talked about with Chinook before. We get the lengths and the gender and whether they're hatchery or natural origin fish. And yeah, and we have to extract the otoliths of every one of these to determine whether they're a hatchery origin fish or not. They when they get released as fry, these sockeye, they're too small to get an adipose fin clip. And so all the sockeye that come back, whether they're a hatchery fish or non-hatchery fish, they all have adipose fins. So we can't tell just by looking at the fish whether it was a hatchery origin fish or not. We have to extract the otolith and look for a hatchery mark on the otolith. And then the sockeye, they're coming through the locks. They come through usually in early July, mid-July is when the run peaks. And they go into Lake Washington in the cooler waters and hang out there for a little while. And they don't start entering the cedar to spawn until late September and early and mid-October is kind of the peak of spawning for them. And then they come up into the cedar and spawn, and then they die, and their carcasses are everywhere in the cedar too. And I just had to share some more carcass pictures. I think these fish are just absolutely beautiful to look at in the fall. The carcasses are. And the previous picture was some male carcasses. These are some female carcasses. And again, like we do with Chinook, we try to cut open the bellies of these female carcasses to assess whether they've spawned successfully or not before they died. This one in the upper left, it only has a few eggs in it. And so this one was probably a successful spawner. The one below it here on the lower left and over to the right, I think that these fish were pre-spawn mortality. So these sockeye appear to have died before they spawned. And we do have a little bit of a problem for sockeye in the Cedar River with pre-spawn mortality. Unlike the Chinook, we get relatively large percentages of the female sockeye in the Cedar River seem to die before they get a chance to spawn, which is kind of sad. And when I say large percentages, some years it's between 30 and 40% of the females that we inspect. It seems like they have bellies full of eggs, meaning that they died before they spawned. So it's kind of a bad problem. We're not sure the exact cause of it. It likely is linked to high water temperatures. Potentially these fish are stressed when they come through the locks in the Lake Washington Ship Canal, and they experience that warmer water, and potentially there's a higher parasite or disease load in the water because the water has been warming up longer earlier in the spring when the sockeye moved through there. And so it's possibly that they come into contact with these diseases as they swim through the Ship Canal, and it doesn't kill them right then, but they go down, and like I said before, they hang out in deep areas of Lake Washington where the water is cool. But if they've contracted that disease, it kind of works on them. And as they move up to the Cedar River to spawn, sometimes they just don't have the strength to complete their spawning cycle. And some of the diseases we've seen, they're diseases that are common in the basin. Well, they're not common, but they've always been around. Columnaris is the name of one disease that has always attacked fish, and Parvicapsula is another one. And these are diseases that have been present in the watershed, but they seem to be acting on these fish more aggressively and at higher rates, and sometimes the fish will have multiple diseases. And so there seems to be a cumulative effect that may be affecting adult sockeye. Okay, so some of these statistics that we get from the carcasses in the cedar, about 62% are natural origin fish and about 38% are hatchery origin fish. And you can see over time, that trend is relatively consistent. So 60% or a little better are natural origin fish, and then about 40 or a little less than 40% are hatchery origin fish. And of course there is a hatchery program on the Cedar River hatchery up at Landsberg. And so that's the only sockeye hatchery program in the Lake Washington Basin. So that's where those fish are coming from. And this is meant to be an integrated population. So the hatchery fish are meant to spawn in the river at low percentages like we've been seeing. And so the ages of these, primarily these sockeye return as age 4 fish. And then in many years, there's a good percentage of fish that also return as age 5 fish. Some return as age 3, but it's usually a low proportion. And if you look at that, those ages over time, you can see that's true in most years. This lightest colored bar represents the age 4 fish, and you can see in most years it's the case that most of the sockeye that return are 4-year-olds, a few 3-year-olds, but not very many. And in some years, the percentage of 5-year-olds is a little bit greater, but usually it's not as great as the age 4 fish. Okay, so that's all I'm going to say about the sockeye. I did want to go back down to the Ballard locks and mention one thing that you may have read about in the paper recently or seen an article about, and it's kind of seal predation at the Ballard locks on many of these salmon species. These are some pictures that were taken by Laura Bogard, who works for Oceans Initiative, which is a nonprofit organization that did a study down there. They actually studied an acoustic device that was designed to scare seals and sea lions away. But the problem is at the locks, it's kind of a constriction point for adult fish as they come back. And you can see at this picture of the left, to enter the fish ladder, they have to enter it through this small door here, or there's another similar door just around the corner. And what we've been seeing, and this has always happened to some extent, but it seems to be particularly bad this year and last year as well, is that harbor seals that kind of live down there year round, they kind of just lurk in this area of the suds, or some of them even go up into the fish ladder, and they're able to corral and catch large numbers of these sockeye and Chinook. It's this fish in this picture that's getting eaten by the seals. It looks like a Chinook to me. But the seals were there throughout the run of Chinook and sockeye and coho, and they really consumed large numbers of these adult salmon down there. So I thought some of you guys would be interested in seeing some pictures of these. Here's some other photos that were taken of these seals, and these are ones that actually did go into the fish ladder. This is one of the first few steps within the fish ladder, and this is during the sockeye run. So you can see these seals these are all sockeye in the mouths of these seals. And in this one picture, you can see two seals are in there together, and they really kind of share the catches, and they seem to teach each other how to catch these fish. But the locks just presents a spot that kind of funnels these adult salmon into small areas and creates opportunities for these predators to catch a lot of them if they want to, and that can be kind of important when we're at lower run sizes like we're at in this day and age. And so I was showing you pictures of seals. Now sea lions always get a lot of attention when they come eat salmon. The sea lions aren't there year round, but they do migrate in. Some individuals do migrate in, and they're also doing the same thing down there. I'm not aware that they're going into the fish ladder as much as the seals. Are so big, it's a little more difficult for them to get in there. But this is a picture of a sea lion just from this summer, and this is during the coho run. So this sea lion had caught this coho and was eating it. One of my technicians that works down there took these pictures of the sea lion down there. And this is this is Laura Bogard herself, and working for Oceans Initiative, and that's a picture of the TAST device. They call it the so that's the acoustic device that emits this noise that's designed to scare seals and sea lions out of the area. And hopefully, in sometime in the future, if this study proves that it works and keeps seals and sea lions away, more of these devices might get installed down there, and hopefully that will be a good solution to the seal and the sea lion problem. What seems to be getting to be a worse problem over the last couple of years for salmon down at the Ballard locks. Okay, so that's my last slide. I think that's all I'm going to say. I think I can take questions now, Charlotte. It looks like I may have talked a little longer than you wanted me to.
Oh,
So.
this is perfect. We had to start a little bit late.
Okay, great.
The link broke in the reminder from Eventbrite. So we started a few minutes late. But thank you so much, Aaron. I love seeing your presentation and your pictures and learning more. I learn new things every year from you, and I'm so grateful. So we do have some questions. Let's see, starting with, we had a few questions about otoliths and hatchery marks. So I'm just gonna, somebody asked, maybe you could review how you tell the difference between a hatchery origin fish versus a natural origin fish. And then there was questions about what is the hatchery mark on the otolith and how do you mark the otolith? So perhaps you could, and how do you get the hatchery mark on an otolith in the fish's brain? So I thought we could start with those 3 questions. Which
yeah. And of course it's different for Chinook and Sockeye. So I'll talk about Sockeye. So like I said before, you can't tell from looking at the fish externally whether it's a hatchery origin fish or not. We have to mark the otolith. And so the way we do that in the hatchery. So when they're, just as they're hatching and as they're fry, you actually, you can change the water temperature. So you chill the water and then warm it back up. And if you chill it for a longer time period, you create kind of like a barcode on the otolith. And so like every day that the fish is alive, that otolith acts like kind of like a tree, and how a tree lays down rings. And the rings are further apart when it's warm and they're closer together when it's cold. And so the rings that get formed in a fish's otolith are just like a tree ring. And so when you cool the water down, that fish every day will lay down these really tight rings, and they'll look like a real dark line on the barcode. And then if you warm up the water a little bit, it'll create rings that are further apart, and it'll look like a light spot on the barcode. And so every year we create this barcode on the otoliths. And so when the fish goes out to the ocean, of course, it looks just like a normal fish. When it comes back, we're able to grind down. We have to grind down the otoliths and look at them under microscopes, and sometimes put a special dye in there. But you can tell that it's a hatchery origin fish from the particular barcode that's put down on Sockeye. And then on Chinook, we don't need to do that with Chinook, luckily. So Chinook, when they hatch out as fry, we hold them in the hatchery and we feed them for a couple of months and let them get to a larger size before we release them. So they're big enough that they can be externally marked. So we do that by clipping off the adipose fin. So you can hold a fish in your hand and see whether it has an adipose fin or not. And so that's how we tell the Chinook. But yeah, I hope that answers the question for otolith marking with Sockeye.
Great, thank you. In kind of along the same vein, somebody's wondering what the ideal ratio between natural and hatchery origins is.
Well, with Chinook, I would ideally you would have for that population in our hatchery program. It is characterized as a segregated hatchery program. So with segregated programs, we would ideally like 100% of those hatchery fish to return to the hatchery, and we would like to see 0% of those fish spawn or stray to the Cedar River to spawn. So we would like them to all. So that is one ideal from the hatchery perspective. Some people really think that it's important to, with low populations in the cedar, like cedar Chinook, that those hatchery fish do spawn successfully on the spawning grounds, and they are locally adapted to this basin. So they do bolster the Chinook population that spawns there, and they probably contribute to higher fry production because of that. So some people see it as a type of recovery measure. Now switching to the Sockeye program, that's an integrated hatchery program. And so the intent is that those hatchery fish will go out into the spawning grounds and spawn naturally with the other fish. And so there, we don't want all of them to go back to the hatchery. We want some to spawn naturally, but we don't want too many to. So I guess in an ideal percentage would be about where we are right now, about 40%, or kind of don't want to go more than 50%. We wouldn't want to see more than half the fish spawning naturally in the cedar be those hatchery adults. And so obviously that's a ratio that's going to vary from year to year. So on average, maybe on a 10-year average, we wouldn't want it to go over 50%. And it hasn't been. It's been hovering around that 40% range. And that's one of the reasons we track that metric. And that's one of the ways that we monitor our hatchery programs in the basin.
Somebody's asking about that now. For many years it was assumed that adipose fins didn't really function, didn't have a natural function, or we didn't know what that function was, is probably a better way of saying that. And now it's suspected that adipose fins are a sensory organ due to this density of nerve endings there. Is anything known about survival rates in hatchery salmon of the same species with or without adipose fins?
That is a good question. We ask ourselves that once in a while. And as of now, I'm not aware of any research that conclusively shows that if a fin or if a adipose fin is clipped off of a juvenile Chinook, that it affects its survival in a negative way. So right now we don't have any real good research that suggests that in any way is reducing the hatchery fish's survival rate. So it could be the case, but the effect would be so small that we haven't been able to detect it at this point. So I don't think so. Is the short answer to that.
Yeah, thank you. There are a few questions too about sockeye numbers and the possible reasons why they might be so low. So I'm just going to read. What are the possible reasons the sockeye numbers are down so much? Problems as they grow to maturity or as adults in the ocean? Or is the problem on their return back into fresh water and towards the river? And then somebody also mentioned last year's presentation for the Cedar River Salmon Journey volunteers. Neela Kendall's presentation, she's a scientist with Fish and Wildlife, who was doing models of fish abundance in sockeye, and wondering if there's any new information. I think the question is about predation from invasive species in Lake Washington that may cause long-term declines in Lake Washington sockeye. And do you agree with those findings? Or are there any other developments that could change this outcome?
Yeah, there's a lot of questions about why the sockeye population is doing so poorly, why it's not doing. But it's all of these salmon populations, they just have a variety of challenges, especially in the lake. I mean, you can see in this photo here, one challenge they have is that this is an urbanizing basin, and stormwater runoff and development. It kind of affects survival at multiple different life stages. But for sockeye, what we currently think is the most limiting factor probably is that when they're juveniles, they have that first year of their life where they go out of the Cedar River as fry into Lake Washington. They spend a full calendar year in Lake Washington rearing as juveniles before they leave out to marine waters as smolts. And so during that year, we think that predation is really limiting their survival rates. So we think that Lake Washington sockeye have lower survival rates as juveniles in the lake than other natural sockeye populations around, like up in Alaska, so to speak. And I think I have some other photos here of, and we think the fish that are eating them the most readily are also, unfortunately, these are native fish that live in Lake Washington. So on the left, you have these are adfluvial cutthroat trout. Those are native to the basin. And then on the right are the northern pike minnow, both native fish species. But these prey pretty heavily in pelagic areas of Lake Washington on juvenile sockeye. And so we think that predation from these species primarily is what is limiting kind of the sockeye numbers. And one thing that's allowed these predators to become more effective in recent years is one thought is the nighttime lighting and kind of sky glow. And it's so at night, the city and the surrounding cities, even Bellevue and Kirkland and Seattle, The nighttime lights shine up and reflect off the clouds. And at night is when the juvenile sockeye that live in Lake Washington, in the day they go into the deeper waters where it's dark and they feel safe down there. But at night they have to come up to the surface because that's where all the food is that they like to eat. And so that used to be the safe time for them to come up into shallow waters and eat. But now the surface of the lake is illuminated, at least like the top 10 or 15 feet of it is all night long. And so some evidence suggests that these predators are able to eat all through the night on sockeye, whereas historically, you know, there was a good 8 or 10 hour time period at night when the sockeye were more able to go up and feed. And then probably another thing that is a big threat for the sockeye is temperature and lake temperature. And so we all know it's kind of been getting warmer, and we have record hot days, and I don't know if that affects the lake temperature as much as what does though is the lake kind of warms up a little bit earlier every year, and it stays warm for a longer time period into the fall. So kind of earlier in May is when we're hearing about these real hot 80, 90 degree days that we didn't used to get, and it really warms the lake up sooner. The lake water stays warmer for a longer time period, and so that's really advantageous to these fish species. These are all the non-native fish species. And they really kind of thrive in that warmer water environment. So we see bass and perch and rock bass and calico bass and even walleye. And so these conditions are ideal for these fish. And when the water temperatures warm up earlier in the spring, these fish, their metabolic rates kind of rev up. They get hungrier sooner, and then they too could start eating more of these juvenile sockeye that are reared in the lake. Whereas historically, these fish were always here, but it was cooler, and these fish kind of go into deeper areas of the lake and become in many senses dormant during the winter. They don't eat a lot, and they don't run down prey, so we don't think that they ate a lot of the sockeye. But as the lake warms up, I think they're taking a bigger chunk and contributing more and more to the predation problem that we think is limiting these sockeye. So yeah, that kind of answers the question for the sockeye, or at least some of our thoughts about what's limiting the sockeye population. So was that did I hit all the parts of the question? Was there a little part that I missed? Or
I think you hit it. Continuing to think about sockeye, did they historically use the Duwamish and Black Rivers to spawn in Lake Washington? We know that sockeye were not historically prominent in the Cedar, but did they use the Black?
Oh, did sockeye salmon use the Black River?
Yeah.
Not to my knowledge. No, there wasn't a big sockeye population, and honestly, there's not really great historical records about the fish populations that went through the Black, the river, but no, there wasn't down there. There may have been a small riverine population, and we still do see sockeye come up into the Green River, and I think they spawn successfully, really small numbers, and they'll rear in the lower areas of the Green and the Duwamish, which kind of acts like a lake for these sockeye, and I think they're able to rear down there, and a few of them make it. But yeah, they back to the original question. I think I see what you're saying. So some sockeye, yes, they did come up through the Black River. They didn't go into the Cedar to spawn. They came into Lake Washington, and they went up into the Sammamish River and spawned in those tributaries there. So Big Bear Creek, Little Bear Creek, North Creek, small numbers of sockeye spawned there historically, probably, and probably a lot of kokanee did too. In fact, some of the historic records from the native tribes that lived along the Duwamish said that kind of the kokanee was one of the favored fish species for them to eat, and they oftentimes preferred those over some of the anadromous salmon that came back. So we know kokanee were abundant in Lake Washington, but we also suspect that there was small numbers of sockeye that spawned in those tributaries, and the ones that spawn up there to this day are probably descendants of those fish. So those are truly native to the Lake Washington basin, the small populations up in the Sammamish River tributaries.
Thank you. We have some questions about ages of the spawners when they return Do we know why a fish comes back at age 2 versus age 4 or 5?
God, no, we don't really. um, I don't know if they, maybe it's all depends on what they experience in the ocean, and it also depends on the survival of that year class. So if in one of those years we find that there's more age 5 fish on the spawning grounds when normally there's age 4 fish, it could mean that there's not really that many more age 5 fish there. That year the fish that were supposed to return as age 4s may have experienced poor survival at some point in their life history. So maybe as they went out as juveniles, when they went out through the Strait of Juan de Fuca or hit coastal waters, there may have been some food shortage, or it might have been one of those the blob years that we talk about, or El Nino, and so that year class may have survived poorly, and so they come back, and that year class is so that's what we characterize as a weak year class, and so then the proportion of fish on spawning grounds that are age 4 fish are going to be lower than normal. And so that's how that is determined a little bit. It's not really so much a conscious decision on the part of the fish. It's how many fish from a particular cohort survive to make it to that spawning phase. So I hope that answered that question.
Yes. Speaking of age, do you track the age of sockeye who have died before spawning? And is there any correlation or pattern between pre-spawn mortality and age?
We've looked at that in some short-term studies, and we haven't been able to find a correlation between age and pre-spawn mortality. So again, most of the sockeye females that we see coming back are in that age 4 year class, some age 5 fish. So usually an age 5 fish will be slightly larger than an age 4 fish, but there is overlap in sizes sometimes. So it's not a huge size difference between an age 5 sockeye and an age 4 sockeye. So maybe the eggs in the age 5 fish might be slightly larger. But there's no known correlation between the age of the fish and whether it's more likely to experience pre-spawn mortality or not. I think it all has to do, the pre-spawn mortality in adults has to do with their exposure to that warm water, exposure to kind of stress along the journey, and maybe potentially what is a higher disease or pathogen loads that may be operating in the Lake Washington Ship Canal or even the lake itself. And so I think it affects all those fish regardless of age, yeah. And regardless of whether they're hatchery fish or not. We see that pre-spawn mortality in fish that spawn naturally in the river. And then we also capture a number of adults and take them to the hatchery where we hold them before they get ripe to spawn. And we see that same pre-spawn mortality in fish in the hatchery. So those are fish that are protected in raceways and have nice water and shade and everything. And they still die at those rates. So I think these fish are exposed to some stressful condition, whether it's water temperature or disease or some combination of the two prior to entering the system or as they enter the system, yeah.
Speaking of pre-spawn mortality, do Chinook succumb to pre-spawn mortality from poor water quality the way coho do on the Sammamish, or it sounds like the sockeye in the Cedar River also succumb for maybe different reasons, but where the question is, do Chinook also succumb to pre-spawn mortality and perhaps from poor water quality if they do?
They do. As I said before, we don't see it that much in fish that spawn naturally in the Cedar River, but some of the fish that spawn in the Sammamish population, we see slightly higher rates of pre-spawn mortality in female Chinook there, not as high as what we see with the sockeye. And then if you go down to the Green River, we see slightly elevated rates of Chinook pre-spawn mortality in the Green River too. And so, all those Chinook, they really experience more stressful conditions than the sockeye as they enter the system. Because if you think about it, they're entering the Lake Washington system right in the middle of August, really. So they got to go through the Ballard locks and swim through that Lake Washington Ship Canal right in August. And that's when it's about as hot as it can possibly be. And so that's kind of a thermal shock to their system. So they have to go through that. But for whatever reason, they seem to be more hardy, maybe because they're bigger bodied fishes. They don't have quite as high as the pre-spawn mortality rate as we see in sockeye. We see a few, but it's much lower, on the order of like 5 to 8 or 10% in Chinook, whereas sockeye can be upwards around 30 to 35% in some years. And then it's important to distinguish the pre-spawn mortality in Chinook and sockeye with coho. So the pre-spawn mortality that people are familiar with in coho happens for different reasons. That's more of a kind of a heavy metal or a chemical in the water that we think comes from stormwater runoff. And so these real urbanized stream systems. And if you think about coho, they could come in as adults during the most advantageous time. They come in later, in late October, or well, at late September, October, they're coming through the locks. And the water temperatures are really quite a bit cooler then. There's a lot less stress for them, and there's higher water flows. And so they should have an easier time from a temperature stress perspective. But they get up into some of these urban systems that have a lot of stormwater runoff that's coming from the road systems. And that is what kills some of these adult coho when they get back into the streams. So we don't see that same type of pre-spawn mortality in coho that go up into the upper Cedar River, for example, and go up above Landsberg and spawn in the watershed. We don't see high rates of pre-spawn mortality in the coho up there. The pre-spawn mortality in coho that's gotten a lot of press are the kind of smaller numbers of fish that spawn in small urban tributaries. So that's a different thing that's killing them than what is killing these the sockeye and the Chinook. So I hope that makes sense.
Yes, it does. Thank you. Speaking of pre-spawn mortality and some of the temperature stresses, do you know if anything's being done to cool off the water in the ship canal? Is that even a possibility?
Yeah, actually some things are. There's been a lot of ideas tossed around. Unfortunately, all the things that you could do to cool water in the Lake Washington Ship Canal are very expensive. One idea that I think is probably the best idea, but also quite expensive, would be to pump water from deep areas of Lake Washington, where it's cold water down there and it has a high dissolved oxygen content. You could pump water up to the head of the Lake Washington Ship Canal. And then the ship canal acts like a river. It's kind of a downstream flow from there out to the locks. And if you pumped a large volume of water up during a couple of months of the year when it was hot, you could create kind of a thermal refuge for the adult fish that are coming up through there. And so there's been talk about that. But again, that's quite an expensive project. Now, the Army Corps of Engineers did try an experiment at the locks. They did what's called a false locking experiment, and there's information about that online if you want to search for it. What that is if you think about it, there's marine water downstream from the locks, and that's highly oxygenated and really cold, so the fish are doing well when they're in that. And what happens when they lock a large boat up is they grab a whole bunch of that cold marine water and they pull it up and they release it up into the ship canal. And it's salt water, so it's not supposed to stay there. It's not supposed to get up into the ship canal or Lake Washington. It's supposed to drain back into the marine waters. And there's actually a special saltwater drain that does that. And marine water is heavier, so it sinks to the bottom and it goes down through that drain. But what the Corps did was they closed that drain, and they did multiple lockages to bring a whole bunch of that cold oxygenated water up into the ship canal during the time frame when the Chinook were migrating, with the idea that would kind of create a cold water refuge along the bottom of the ship canal. And they tried that for a couple of different years, but what happens is that heavier water, the saline water, goes to the bottom and it stays cold and oxygenated for a while, but because it doesn't mix with the upper water, it loses its dissolved oxygen and it becomes anoxic over time, which is bad. It kind of kills some of the microbes in the bottom sediments. And what happens is that saltwater wedge, in those years they did the experiments, it crept upstream and actually filled up the bottom area of Lake Union. And then it actually went up past out through the Montlake Cup and started to spill into Lake Washington, which is something we don't want to have happen. And so they kind of determined that maybe that was a good idea, but I don't think it seemed like a good way to fix the temperature problems in the ship canal. So we kind of tried that and abandoned that as an idea that wasn't the best. So there's a few things that I can tell you about what could be done for water temperature in lakes in the ship canal. Yeah.
Thank you. Well, I know it's 7:49 now. We still have a few questions, but I also want to be sensitive to your time and the time of our participants. So do you prefer that I read out the questions now, or we could email them to you and get answers later? What's your...
Oh, like 2 or 3 more? Yeah, we could push through a few more. Yeah, okay.
Yeah.
Wow, sure.
How about we'll stay on for about another 5 or 10 minutes and then we'll let you go.
Yeah, that sounds great.
Okay, and then we'll follow up with any unanswered questions via email.
Yeah.
Thank you. Let's see. Okay, so can you tell whether a wild Chinook hatched in the Cedar or the Issaquah area rivers?
You can't really tell now if it's unclipped. One way you may be able to tell is to extract the otoliths and do what's called otolith microchemistry. And if kind of the elements in the water on the Cedar were different than the Sammamish, and you were able to detect those from the otolith microchemistry analysis, that would be one way to tell if that fish originated from one watershed versus the other. But I'm not aware that the geology is different enough in the Sammamish River tributaries from the Cedar River for one of those analyses to be able to conclusively tell you one way or the other where that fish came from. So I'm gonna say it would be difficult to know. Yeah.
Okay. Why do Chinook from Issaquah Hatchery stray to the Cedar River? What does that tell us about salmon going back to spawn in the place they were born?
So yeah, that's a good question. So here's one theory that's been around, and nobody knows this for sure. So if you looked at that original map that I put up there, I won't pull it up again, but the headwaters of Issaquah Creek, which is where the Issaquah Hatchery is, the headwaters of Issaquah Creek are very close geographically to the headwaters of the Cedar. Actually, they come out of the same hillside, but there's they just go down different paths. And so when salmon come into freshwater, they start using their olfactory system. They cue into the different smells of the different water, and then people think that the water that's coming through the Issaquah Hatchery smells very similar to the water that's coming out of the upper parts of the Cedar River watershed, and so that may influence the number of fish that kind of stray, so to speak, up into the Cedar River. And if you look at it, we don't see a lot of the hatchery fish straying into the lower parts of the Cedar, but if you look at the fish counts at Landsberg, we see a little bit of a higher proportion of hatchery fish going up above Landsberg. So when the hatchery fish enters the Cedar, it really seems to want to go as high in the watershed as possible, which would support that theory that they're kind of homing into a geochemical smell or cue in the water that they think is where they're supposed to go. And it's just coming down the Cedar River instead of down the Issaquah. So that's one theory that's been around for a long time, why Chinook would stray there versus going back to the hatchery. And then I don't know if you saw, we used to have relatively low stray rates into the Cedar for Chinook, but they've kind of increased a little bit in recent years. And one thought that's been around there is there again, it may have to do with the increasing temperatures in Lake Washington and increasing temperatures in the Sammamish River. And so during the time frame when hatchery Chinook need to migrate up through the Sammamish River to get to the hatchery, the water's really hot in the Sammamish River. It's a terrible place for Chinook to have to migrate through. I'm surprised as many do it as we have happened, but one thought is that some of the Chinook kind of nose into the Sammamish River and say, gosh, this is too hot, and they mill around and go back down, and they find the Cedar River, and they think the water is cooler there, which it is. And so that water temperature is what exasperates the stray rates into the cedar. So that's another thought. But to be honest, I can't say for sure. Those are some ideas that have been tossed around, but I don't know for sure.
Are offspring of hatchery fish considered wild? Are they physiologically the same as wild fish? And do they return to the same stream that they were, well, born in?
Yeah, they would imprint on that area where the red was dug and where the egg was incubated, and as they hatch out and migrate down that stream, that's when they imprint. And so yeah, you would think of that as a first generation hatchery fisher, but I would consider it a wild fish. And we try to wild fish is we try to shy away from that term sometimes. We call it a natural origin fish. So yeah, its origin, it originated from the gravel, from a naturally spawning pair of fish in the river versus hatchery origin. So to say that fish is wild, yeah, there's a lot of definitions for it to be a true wild fish. A lot of people say that has to have no genetic genealogy in its ancestry. And so that fish, if you did a genetics test, you could link it back to a hatchery program if it was a segregated program. And so it wouldn't be a true wild fish in many people's eyes, but it is a natural origin fish because it has been spawned naturally in the gravel. And, yeah, so
when the school groups are releasing their fish that they've raised in the classroom, those would be considered wild when they return.
Yeah, they would be, I think. Yeah.
Oh, sorry, natural origin.
Natural origin is what
Yeah.
I would consider.
Yeah.
They would come back. They would not have, and most of those are coho. They would not have a fin clip. Yeah, and they would home back to that stream in theory from where the fry were released. So I would consider those natural origin coho. Yeah.
Okay. Well, it looks like we're about out of time. There are a few other questions that haven't been answered. So I'm just going to check in with Danny to make sure we can record those questions and get them to, and so we can email these out to the group who attended tonight. And so Aaron, I'll be following up with some questions. There were a few questions about seals and predation and how the new device might be affecting salmon as well as the pinnipeds. So
oh yeah,
a few questions about that, and then a question about like, where was the picture of the sturgeon taken? Because that's so cool.
Oh, right. Yeah, those
yeah.
sturgeon are cool. Yeah.
So I will email those to you. One quick question. What is the most rare salmon? That's a trick question.
Oh, whoa. In the world or in the
no, I don't know. They didn't qualify, but maybe in the Cedar, since we are based on Lake Washington and Cedar River.
Oh gosh.
There's
well,
one we might see.
Yeah, going by the numbers, maybe the Chinook are the most rare salmon.
Yeah, definitely. They are threatened in the Cedar River, so.
It is a threatened population. Yeah, so maybe you'd have to go the numbers wise, they're the lowest. Even though the population is relatively stable, I would say maybe that's the most rare kind of the special one.
Thank you. So somebody's also asked if you are willing to share your slides, and you have very generously given permission for us to share this presentation. So we will post it probably to the Aquarium's YouTube channel, and we will let our attendees know the link to that, so anybody can rewatch or anybody who wasn't able to attend can check out this fabulous presentation that we learned so much from. So Aaron, thank you so much. I know this is a very busy time of year for you. You're out there floating the Cedar River on Tuesdays, as well as other days, it sounds like.
Yeah.
So yes, I so appreciate you taking the time to put this together for us tonight. And good luck, and I hope you have good weather and lots of wonderful salmon sightings out there. And we look forward to hearing about what all your data suggests for this year's return to the Cedar River and to the Lake Washington Basin.
Okay.
Yeah. So thank you. And then everybody who is on. There was one question about where's the best place to go see salmon right now. I would say the Cedar River. Aaron, do you have anything else to suggest before we say goodnight?
Always the best place is right at the Issaquah Hatchery. If you've gone there, there's that little bridge and you can look down, you can see a lot of Chinook spawning naturally down there. Just a great place to see spawning Chinook. But yeah, other than that, the lower Cedar, right along Renton, go take a walk on that bike path through Renton that goes along the river, and you should be able to spot some sockeye down there spawning, or at the fish collection weir right under the I-405 bridge, which is also along that bike path. Those are good places to go. You can see fish there.
Yeah, check out the Seattle Aquarium's web page for Cedar River Salmon Journey program, and we have a map there for great salmon viewing locations along the Cedar River. And email us too at salmonjourney@seattleaquarium.org if you have any questions or want some suggestions about where to go see salmon. So again, thank you so much, Aaron, and thanks to everybody for joining us this evening. And we hope to see you next Tuesday for our final session, where we'll learn about what is being done to help some of our struggling salmon populations in our local watershed, the Cedar River and Lake Washington and Sammamish watershed. So until Tuesday, thank you and goodbye.